Biology, Part 215. Even when it is present and similar in size, the Y chromosome very often seems to be genetically inert, carrying few, if any, functional genes. By contrast, the X chromosome has many loci carrying the range of important genes. XX individuals have two alleles for these characteristics, but XY individuals have only one. The figure shows the result of two different crosses involving red-eyed and white-eyed Drosophila. This characteristic is controlled by a gene carried at a locus in the X chromosome, XR, in allele of the red eyes, and the dominant in XR, the allele for the white eyes. In the first cross, the red-eyed female parent is homozygous dominant. All her gametes carry the XR allele, which is thereby transmitted to all the offspring. The white-eyed male parent with the both X and Y chromosome possesses just one eye color allele located at the X chromosome passed to the X-bearing sperm. As you can see, only the female offspring receives X in the allele for white eyes, making the heterozygous excess. Since X is dominant, all the offspring's females show the red-eyed phenotype. Chromosome abnormalities. Non-disjunction. Number one is non-disjunction. The normal process of meiosis ensures that each gamete will contain a haploid set of chromosomes. For example, human gamete contains 23. Sometimes, however, one of the male pairs of homologous chromosomes fail to separate, so that some gametes contain extra chromosomes while others contain too few. Not disjunction of the sex chromosomes causes two principal types of the genetic disorder called the Klinefelter syndrome and the Turner syndromes. In plants, non-disjunction sometimes affects not just a single pair of homologous chromosomes, but all pairs simultaneously, with the result that gametes containing a full diploid set of the chromosomes are formed. The fertilization involving such gametes can then produce the triploid tercourse. Number two, the chromosome mutation. The chromosome mutations and in the sequence of genes carried, although not in the genes themselves, the gene mutation is a separate process. As described in our tutorial, changes are most likely to occur when the chromatids break and rejoin during the crossing over in phase 1 of the meiosis. As a result of the section 1 chromosome may be lost altogether, they incorporate into another chromosome. A number of specific abnormalities are possible, outlined in the figure. The deletion involving in the less of the genes is often fatal. However, inversion, the translocation, and duplication all lead to the reshuffling of all the genes. Extranuclear genes. The Mendelian inheritance mechanism operates when the genetic material is carried on homologous pairs of the chromosomes in the nucleus of the eukaryotic cells. However, not all of the cells of the DNAs are confined in the nucleus, and we go through them, and a small but significant proportion is present in the cytoplasm inside the cellular organelles. The mouse liver cells, for example, of the mitochondrial DNA represent about 1% of the total, while the leaves of the maize as much as 15%. The organelles and their genes are transmitted to the offsprings in the cytoplasm of the gametes. Obviously, the gametes are similar in size. Both parents will contribute extranuclear genes, but if they are very dissimilar, such genes may come from only one parent. Mammalian sperm cells and the pollen grains from flowering plants contribute very little cytoplasm to the zygote, so that the inheritance of the Organ cells is often completely maternal, so it goes along with it.